My name is Natalia Tretyakova. I am a distinguished McKnight University professor at the Department of Medicinal Chemistry and the Cancer Center at the University of Minnesota. We are fortunate to have NIH funding for 5 major projects. The main project that has traditionally been the focus of my lab is investigation of the chemical modifications of DNA. So what we do in this project is we use organic synthesis to synthesize non-canonical DNA bases and nucleosides that are formed as a result of chemical exposure. We then use solid phase synthesis to generate small pieces of DNA containing these unnatural nucleobases. We then use the tools of molecular biology to incorporate the synthetic DNA into cellular DNA. Then we can work with cell culture to investigate the outcomes of these chemical modifications in cells. So this work enables us to understand the chemistry of DNA damage and also the mechanisms of how it affects the cellular function, such as DNA replication, transcription, and to establish repair mechanisms of such damaged bases. In the second project, we're focusing on the origins of inter-individual differences in cancer. So, for example, in people who smoke, there is a large difference in lung cancer incidence between different ethnic groups. African American smokers and Native Hawaiians are at much higher risk for development of lung cancer as compared to Latino and European Americans. So we hypothesize that these differences in lung cancer risk originate from genetic and epigenetic differences that are observed between these ethnic racial groups. So we are using the tools of analytical chemistry and epigenetics and also genetics, such as genome-wide association studies, to understand how these different ethnic groups metabolize tobacco carcinogens differently, leading to much higher risk in specific groups of individuals compared to others. This is very important because we want to understand which smokers are at high risk for the development of lung cancer, so that these individuals can be targeted for smoking cessation and chemoprevention therapies. In the third project, we are focusing on epigenetics. This is a relatively new area in my laboratory. We just published the first big paper in Nature Scientific Reports. And there we investigate DNA methylation and demethylation. DNA methylation occurs primarily at cytosine bases of DNA and is used to silence genes so that the patterns of gene expression can be established. So we know what proteins introduce methylation marks. Those are DNA methyltransferases. But for many years, it wasn't known what proteins remove the epigenetic mark, this methylation mark from DNA, enable us to turn the genes back on. So it was found that 1011 translocation dioxygenases play a very important role in oxidizing the methyl group within DNA to hydroxymethyl C, formyl C, and carboxyl C. And then these oxidized forms are recognized as DNA damage and are removed by DNA repair machinery. So this enables these genes to be activated by removing a repressive mark. So we are focusing on Tet proteins. There are actually 3 of them, Tet1, Tet2, and Tet3. And these are metalloproteins that use alpha-ketoglutarate cofactor to oxidize the methyl groups in DNA. So we want to understand the roles of these oxidized bases in DNA by conducting chemical proteomics experiments to identify specific readers of this hydroxymethylated, formylated, and carboxylated cytosines in DNA. And also we are looking in animal models to understand the effects of inflammation on epigenetic marks. So we have a large grant from the NIH to understand how epigenetics provides a link between inflammation and cancer. In our fourth project, we investigate DNA protein cross-linking. So in the presence of bis electrophiles or free radicals, proteins can become irreversibly trapped on DNA. So we are interested in this process because formation of such DNA protein crosslinks appears to be associated with aging. So we know that as we age, specifically in the heart and in the brain, there is an increase in the number of DNA protein crosslinks. So my project, that is actually a collaboration with Colin Campbell in pharmacology, it aims to understand how this DNA protein crosslinks form and how they're handled by cells, and how maybe when this repair process fails, it could lead to aging and other diseases. Very recently, with the COVID pandemic influencing the world, we also became engaged in investigation of potential inhibitors of COVID RNA polymerases. So our lab is known for our ability to synthesize unnatural nucleosides. And so we are making analogs of remdesivir, which is one of the therapeutic agents that's currently being investigated for treatment of COVID. And we are using the tools of molecular modeling, organic synthesis, and also biochemical assays to test the activity of these analogs in human cells. So what kind of links these different projects is our expertise in nucleic acid chemistry and also in mass spectrometry. My laboratory has two powerful mass spectrometers. One is a Quantiva triple quadrupole mass spectrometer, and the second one is a Q-exactive Orbitrap mass spectrometer that allows us for accurate mass determination and very high resolution analysis of biomolecules and small molecules. So each student who joins the lab has the opportunity to learn hands-on how to operate this instrumentation, and all of them apply mass spectrometry in their research. My laboratory is probably a right fit for someone who wants to use the tools of chemistry and biology to be able to achieve a specific research goal. So we don't limit ourselves to just making a compound or just doing the biological studies. We are able to combine these two so the trainee can experience organic synthesis, solid phase synthesis, work with cell culture in animals, and also be engaged in human studies, all in one project. I think that's pretty unique, and so if that's something you are interested in, I hope you'll consider my group.